like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the National University of Singapore Yong Loo School of Medicine Healthy Longevity webinar. Thank you for joining us, and especially your interest in healthy longevity, of course. We have a great program for you today. Professor Manuel Serrano is with us tonight, and I will introduce him in a few minutes. First, I would like to remind you to use the Q&A function for your questions, which we are going to address after the presentations, because first we have a short presentation and we'll hear from Gua Willi, a senior postdoc at our Center for Healthy Longevity and the Center for Reproductive Longevity and Equality at the National University of Singapore. And she has very interesting results, her own findings she is going to report on, namely how small amount of stress can drive proper mitochondrial function, and therewith potentially in the future can slow down aging. The floor is yours.
Right. Hi, everyone. As you've heard, I'm Hui Li. I'm a Senior Research Fellow at the US Center for Healthy Aging and the Center for Reproductive Longevity. And today I'm going to tell you about a scenario when some stress can be beneficial. Now, when we think about mitochondria and aging, this is what we normally think about. It's a simplified diagram of a cell here, and you have the nucleus where DNA is stored. You have the mitochondria where the bulk of the energy is produced in the cell. Now, during the process of producing energy, mitochondria also makes reactive oxygen species. Now, you can tell from the name, reactive, right? So this reactive oxygen species, or ROS, they can also react with DNA and cause damage to the DNA. So normally we take ROS as bad. Now, but we know that as cells age, their mitochondrial function is less and less good. And so what we wanted to do is to find out what is needed to keep mitochondria functioning well, and how do we keep making mitochondria. So we use a brown fat system, starting from cells, and then we turn them into mature brown fat cells. Brown fat cells are brown because they contain lots of mitochondria. So during this whole process, lots of mitochondria are being made. And then during this process, we collect the cells and we measure how they make proteins, and we measure how they make proteins in different parts of the cell. Now, the making of proteins is known as translation. And in essence, this is basically how you translate the code in DNA into the making of proteins into amino acids, the building blocks of proteins. So for example, here, this is literally the code of life that you see. The code is triplet. Right, it takes the form of 3 letters. So in this case, CCU codes for this amino acid called proline. Now, proline is encoded by 4 possible triplets or codons. So the code is redundant in that sense. Now, we did our experiment and realized that even though, for example, for proline, you can have 4 possible triplets to use, during this brown fat differentiation process, when they make lots of mitochondria, only 2 of the codons are preferentially used, which is CCC and CCG. And this occurs for many of the amino acids depicted here. So basically, there's a translation bias towards codons that end in G or end in C. Now, and this signal, this translation bias, starts from the mitochondrial vicinity and then spreads to the rest of the cell. And we figured out that the signal is actually reactive oxygen species that's produced from the mitochondria, which can then later spread elsewhere. So how does this matter to the cell, and how does this drive brown fat differentiation? So we know the sequence of all the genes in the cell, so we can figure out which genes tend to overuse GNC ending codons and which genes underuse. The genes that overuse GNC ending codons would benefit from the translation bias that we see. And we can figure out that it's the mitochondrial membrane components as well as brown fat regulators that tend to overuse triplets that end in G or C. So we know that once you have this translation bias that starts, then your cell makes more mitochondria and they become more brown fat-like, which is good. Now, a group of genes that we were not expecting, but which showed up, were the Ros Scavengers. These are proteins that sponge up the ROS present in the cells, so to reduce the amount of ROS present. So instead of thinking the conventional thinking that all ROS is bad, here we have a scenario where you need some initial mitochondrial biogenesis that makes some ROS as byproducts, and then this triggers a translation bias, and that benefits the translation of certain genes, including the Ros Scavengers. And then this serves as a negative feedback loop to go back to the initial bits of mitochondrial biogenesis that to dampen down the overall levels of reactive oxygen species produced so that the cells are not damaged, but they can still sustain lots and lots of mitochondrial biogenesis. Now, this is reminiscent of this process called mitohomeosis, which is a recently discovered phenomenon where you need a certain level of stress on mitochondria in the form of ROS that then leads to increase in lifespan and health span due to improved metabolism, for example, by better mitochondrial function. And so to put the two what we observed together with mitohomeosis. So here you go, brown fat differentiation, you have this diagram that we drew before. And then for mitohomeosis case, you also have a case where you need some initial ROS that then triggers more mitochondrial biogenesis. So it could very well be the case that this translation bias that's triggered by ROS that we observed also takes place in mitohomeosis. And in other words, it is not as straightforward as how we normally think of it, when once you take lots and lots of antioxidants, you normally improve lifespan and health span. So that is not necessarily the case, because there are cases where you do need a certain level of stress in the form of reactive oxygen species to kick off some process, for example, mitochondrial biogenesis. Right. So I would like to thank the people who contributed to the work and funding sources. Thank you very much.
Thank you for the great talk. And now we know that sometimes a little bit of stress is not bad. Now over to our next speaker. It's my really great pleasure to introduce Professor Manuel Serrano. He works in lovely Spain, in Barcelona, at the Institute for Biomedical Research. And he established there in 2017 the Cellular Plasticity and Disease Group within the Molecular Medicine and Research Program. Hopefully that's right, Manuel. He is known for his contribution to understanding lots of pathways being involved in the aging process from different perspectives. And if you see his CV and all the publications, you can see publications in cancer patients and metabolism and regeneration. And not the least, he is also the president of the International Cell Senescence Association, ICSA, where we met at a certain time, I think 10 years ago, and we are working together in the steering committee. It's my great pleasure, Manuel, to have you here on the show, and we are really looking forward to hear your talk and then have a little bit of a chat.
Thank you very much, Andrea. Thank you very much for this invitation to this prestigious series of lectures. Thanks to everybody that is listening. This is a great opportunity for me to tell you about what we are trying to do about reprogramming, using reprogramming for rejuvenation. This is a new area of research, a young area of research. There are many open questions, but it's very promising, and I hope you can get the same feeling that I do about the potential of this. I'm going to start sharing my screen. This is the title of my talk, Understanding and Manipulating Cellular Reprogramming in Vivo. A few words about cellular reprogramming. Let's start with the process of differentiation. I think most of you are probably familiar with this cartoon. This is the Waddington landscape that basically says that during development, pluripotent embryonic cells make a series of decisions that are irreversible, basically, until they reach the final stage of differentiation, of full differentiation. This is a unidirectional process, fortunately, although there is an emerging field of plasticity. And this plasticity is usually manifested when there is a damage in the tissue. This is something that I would also like for you to keep in mind, that the natural capacity of cells to be plastic is triggered when the tissues are damaged. Damage triggers plasticity. The microenvironment of damaged tissues triggers plasticity. But reprogramming is more extreme. Reprogramming using the Yamanaka factors consists in turning the cells to the top of this hierarchy. So adult somatic cells are transformed into embryonic pluripotent stem cells. And this transformation is achieved by the simultaneous expression of 4 transcription factors, OCT4, SOX2, KLF4, and MYC, that I will refer to them as OSKM. There are other combinations, but this is the canonical combination. This canonical combination of transcription factors is able to hijack the transcriptional machinery of the cells. No matter what type of cells they are, they will become transformed by these 4 transcription factors. The configuration of the chromatin will change, and they will become embryonic pluripotent stem cells. There are still many things that are unknown about how this transformation happens, but most people working in this field, they believe that is not development in reverse. So the route to go from here to here is not going upwards in the Waddington landscape, but it is a different route, although this is still debatable. This is a summary, very simplified, of what is known about reprogramming. When these 4 transcription factors are introduced into adult cells, usually there are 3 steps that are distinguished. There is a dedifferentiation. This is the first thing that happens. And actually, these transcription factors that are usually positive transcription factors, they do very little in activating transcription. Most of the job that they do initially is to repress transcription. They repress the host cell identity programs. So the cells, they lose their identity, and they become something that is very poorly defined, something that is very diverse. And this is what I try to convey with this funnel. The variety of cells becomes very diverse. And many of the cells are abortive, many of the cells die, many of the cells become senescent, and many of the cells become identities that probably are not physiological. But some of these cells are able to activate. Now these factors activate. So here they repress, but now here they activate. They are able to activate the pluripotency embryonic program. And they will go through this point of no return. And at this point, the process will be automatic. If the process of reprogramming is interrupted here, many of the cells will disappear or die, but many of the cells will go back to the original identity. And this is important. Many of the cells will return to the original identity because they maintain epigenetic memory. It has not been completely removed. So what happens in vivo? This was our main contribution to this field. We generated mice where it is possible to activate the 4 Yamanaka factors. We call them Yamanaka factors because, of course, maybe I didn't say that, but Shinya Yamanaka was the scientist who discovered the potential of these 4 transcription factors to induce embryonic stem cells in adult cells. So we generated these mice where we can induce in a ubiquitous manner the expression of the 4 transcription factors. We are now doing cell type specific, but I have to say that this is a bit more complicated than we anticipated, because the Yamanaka factors, the first thing that they do is they block the identity, the cell identity genes. So we enter into this loop when the Yamanaka factors, they silence the cell identity genes. So we have to go around that. All the work I'm going to show you is with this ubiquitous expression of the Yamanaka factors. And actually in mice, in adult tissues, we can observe remarkably the same events. The differentiation in some cells, not all the cells they differentiate. Very diverse mixture. We are characterizing this with single-cell RNA-seq in vivo. And there is a point of no return. When we have gone through this process, this takes about 10 days, not very different from in vitro reprogramming. So after 10 days, if we maintain the doxycycline to activate the transcription factors, the mice will go through this point of no return. So now if we switch off the doxycycline, these cells that have arrived to this point, they will be able to form teratomas. And so this is the most visual consequence of reprogramming, teratomas. Some people got disappointed and said, oh, the Yamanaka factors cannot be used for regeneration. Well, let's wait for that. It's true. If we go through this point of no return, they will produce teratomas. But for us, it was good news because this was the way to convince the community that we were actually producing embryonic stem cells in vivo because they produce teratomas. This is the evidence, unquestionable, that there is full reprogramming in vivo. Let me walk you through the pancreas, because the pancreas is the tissue that in our mice reprograms the fastest and the broadest. Is more cells undergo this process. And I always get the question of why, so I will answer that now. We know that damage, tissue damage, is an important component, it's an essential component. So the expression of the Yamanaka factors produces tissue damage, and depending on the degree of tissue damage, we have more or less reprogramming. For example, there are tissues like the lung where there is no tissue damage and there is no reprogramming. However, if we artificially damage the lung, now the Yamanaka factors can produce reprogramming. I'm trying to connect this with what I said in the very beginning, that normal tissues have some degree of plasticity that is triggered by injury. So this degree of plasticity is required for the Yamanaka factors to produce reprogramming. I will go back to this at the very end of my talk. So the pancreas is super sensitive to damage. There is a lot of tissue damage and there is a lot of reprogramming. During the first 3 days, we don't see anything at the histological level, but at day 6, we start to see pockets of cells that look abnormal. They coincide with some cells that are senescent, they are damaged. They're like, we see that with P21. Not all the cells, of course, but some of them are positive for P21. And for us, this is an indication that there is a tissue damage in this microenvironment. We don't see nanog. We have to wait more days to see bigger focal lesions of dysplasia. We call this dysplasia. Now we have even more damage, and we start to see some cells, a few cells, that start to express nanog. Nanog is a protein that is uniquely expressed in embryonic stem cells. So now we have here an adult mouse where we are able to reactivate the expression of a protein that is characteristic of the pre-implantation embryo. So we are going back all the way in development. This is the point of no return. If we eliminate doxycycline here, the process will continue. These plastic foci become bigger. The senescent cells are eliminated to a large extent, and these foci of nanopositive cells become bigger, and they, in a month, it will take about a month for these foci to produce a full-blown teratoma with all the characteristics of a teratoma. This is to illustrate this variety, this diversity of cells. This is an adult pancreas, and these are the surviving, remaining, normal-looking acinar cells. However, this is in a sea of abnormal cells, cells that have different morphologies. And if there are aficionados here to the pancreas, let me tell you that this is not acinar ductal metaplasia. It looks like, to some extent, but it is not. We have characterized this by RNA-seq, comparing acinar ductal metaplasia with this dysplasia. Yamanaka factors-induced dysplasia. Okay, so what happens in other tissues? Well, in other tissues, we see something similar, that the kinetics is more delayed. This is, for example, in the kidney, the tubules, you can see the loss of E cadherin. So the cells are losing their identity at this point. Cells are also losing their identity in the colon or in the stomach. And as I said, in some tissues, we need to exogenously produce a damage, and then we will see this process of reprogramming, and this is what happens in the lung. Okay, so this has been the introduction to in vivo reprogramming, and now why is this interesting for rejuvenation, that I think is the main interest of the audience? Yamanaka, in the first paper that he published, he already indicated that the process of conversion of somatic cells into pluripotent cells could be a way of rejuvenating. It was an open question, but he speculated that it could be a rejuvenation. And actually, very soon after that, we and others started to study the clocks that were known at that time. At that time, we didn't know about the DNA methylation clock, but we knew about the P16 clock. P16 is a protein that is expressed at increasing levels with aging. So all mice express high levels of P16, young mice express almost undetectable levels, and embryonic stem cells express 0. So we reported that in this process of reprogramming, the cells in vitro, the cells reset the P16 clock to the embryonic state. This P16 clock, of course, is not as accurate as the telomeric clock, and the telomeric clock is not as accurate as the DNA methylation clock. The DNA methylation clock is now known to be reset to 0, and because it is so accurate, Stephen Horvath and his collaborators, actually, when they measure the age of the IPS, of the reprogrammed cells, it is not 0, it is -9 months. They have that kind of precision. So somatic cells, they go back to this state. And now the question is, what happens when these cells are used for differentiation? Well, the clock starts from 0, it starts to tick. Other investigators started to speculate about the possibility of not changing the identity of the cells. Maybe, of course, and this is the problem of the teratomas. If we change the identity of the cells, we have a problem. The cells get confused, the cells will produce teratomas in the pancreas, and we don't want that. We want the pancreas, the old pancreas, to remain a pancreas, younger, but still a pancreas. So the concept began of doing partial reprogramming, going close to the point of no return, switching off the Yamanaka factors, OSKM off, and now the cells, many cells retain the memory, the epigenetic memory, and they go back to the initial identity. So in this process of partial reprogramming and reversion, cell identity is not changed. We start with pancreas and we finish with pancreas. And the question is, are these cells rejuvenated? Well, now I have to enter into the world of what do I mean by rejuvenation? Because when we talk about an animal, there are good proxies and good measurements of rejuvenation. But when we talk about cells, it's more ambiguous. We are talking about cellular features of age. So the methylation clock is gaining a lot of attention because of its accuracy. But please be aware that the relation between the methylation clock and transcriptional programs is unknown, and probably there is suspicion that there is no connection. The transcriptomic clock is different from the methylation clock, and the connection between this methylation clock and cellular fitness and tissue fitness and organismal fitness is correlative, but is not well established yet, the causal connection. This is a work of Wolf Rake that is in bioRxiv, where they interrupt the process of reprogramming at different points in vitro. These are human cells of about this age, methylation age. When they initiate reprogramming, many cells are unsuccessful, as it happens in vivo. Are these cells? They don't rejuvenate, they don't reprogram, they don't change, they don't lose differentiation markers. However, the cells that lose differentiation markers, they become younger according to the methylation clock. And this intermediate age is stable. They can go back here, and they, to some extent, they retain the younger age. If they change the identity to IPS, the cells go back to 0 or to -9 months. I don't know. Okay, I have 6 minutes. Okay, but it's okay. So we have been studying this. What happens with the clocks in vivo? So we do partial reprogramming and rejuvenate and recovery. And what happens is that there are no teratomas because we don't go through the point of no return. Tissues recover, and tissues, they look normal. I have to mention here the pioneer work of Belmonte, Juan Carlos Izpisua Belmonte, Vittorio Sebastiano, and David Sinclair in 2020. I forgot to add here the date, the year. They have shown that expression of OSKM in vivo can rejuvenate the cells, but it's a partial expression. There is no change of cellular identity. This is without changing cellular identity. We have been doing the same with a single cycle of OSKM in naturally aged mice. And I will go quickly through this. This is a transcriptomic clock. We did RNA-seq, separated young from old, and asked where are the old mice that have been reprogrammed. And I want to insist in this 2 weeks of recovery. And you will see why I say this. The transcriptomic clock is in between. I will go quickly, and this is important. The signature that most clearly changes with reprogramming is the Mtor signature. I think that this is important, especially in your institute. The Mtor signaling is upregulated with aging, that was known. And when we activate the Yamanaka factors in old mice, they become similar to the young mice. In this signature is the one that rejuvenates the most. We have done also with Wolf Rake a methylation clock. These are individual mice, principal component analysis, old mice, young mice, and the mice that have been rejuvenated are in between. This is only 1 week. This is not many cycles. It's just 1 week in mice that are old. I'm going to skip this because maybe it's not so interesting, and I want to focus here. We have been studying how many of the methylation changes happen immediately after OSKM expression and how many happen during the recovery process. To our surprise, many of the demethylation changes happen after OSKM has been switched off. This was a surprise. So this demethylation is not happening directly by the Yamanaka factors. Actually, when they are switched off, in the process of reconstituting the chromatin is when demethylation happens. In the case of remethylation, and this is here, most demethylation happens after, in the process of recovery. However, the gain of methylation that happens in other sites of the clock, most of the remethylation happens during expression of the Yamanaka factors. I think this is telling us something about the mechanism because, as I said, almost nothing is known about the mechanistic basis of the methylation clock. So this is one piece of information. And we have validated that, well, this is another important thing. About half of the methylation changes that are produced directly by the Yamanaka factors disappear in 2 weeks. We have to think that these Yamanaka factors are acting on a small subset of cells. As soon as we switch off the Yamanaka factors, these cells remain in the environment of an old mouse. And these soluble factors are known to have an effect, and probably some of them accelerate aging in these younger cells. So this is an important thing to keep in mind, how stable are the changes. Most remethylation happens during expression of the Yamanaka factors, and most demethylation happens afterwards. And we have here examples with single CPGs measured by pyrosequencing. We measure how after, this is the time of, this is all mice, this is 2 weeks of recovery and 4 weeks of recovery. The longer the recovery, the longer is the deepness of this demethylation. And I'm going to show you here, just to finish, one example where we have done cycles of reprogramming. I mentioned that Belmonte and Sebastiano and David Sinclair have done this in different tissues, in the retina, in the pancreas, and in the muscle. We have done this work in collaboration with a neurobiologist here in Spain, and we have studied the brain. Adult neurogenesis is the first feature of aging in mammals. As soon as 6 months of age or 10 months of age, mice start to lose the capacity to generate de novo neurons in the suprabasal layer of the dentate gyrus. The same happens with humans, of course, at later ages. It's the first physiological process of aging. The amount of these neurons is directly correlated with the capacity to memorize. So what we have done is we have activated the Yamanaka factors in cycles that are shown here from 6 months to 10 months. And what we have observed is that doing this, we maintain the amount of adult neurogenesis in mice that are 10 months old. At 10 months of age, mice have already lost a lot of neurogenesis. These mice preserve higher levels of adult neurogenesis. Because they have more adult neurogenesis, they are expected to, I want to emphasize, we are not inducing neurogenesis with the Yamanaka factors, we are preserving the young neurogenesis in these mice up to 10 months of age. And because they preserve neurogenesis, they have better capacity of long-term memory. This is one typical assay where the mice are exposed to 2 objects, then one of them is repeated. The mice don't pay attention to the old object. They go to the new one. They remember that this is known, and they do this task equally after 2 hours. However, if we wait 5 days, now the control mice, they don't remember that this was an old known object, and they pay equal attention to this and this, 50% to each of them. However, these mice, they remember that they had seen the cube, and they pay attention most of the time to the pyramid. So this is a physiological impact of these cycles of transient reprogramming. And with this, I'm going to go to the last slide. I'm going to go super quickly here and go to the last slide because I want to tell you what we are trying to do. Our goal, our dream that we are far to achieve is to be able to produce this transient reprogramming without the Yamanaka factors. We want to do Yamanaka factor transient reprogramming without genes. And for that, we want to learn how to improve reprogramming in vivo, how to boost it with pharmacological or nutritional interventions. So for that, we are studying drugs, we are studying the microenvironment, and we are studying the nutritional requirements. And some of this is published, like recombinant IL-6. If we inject recombinant IL-6, we improve reprogramming. If we give palbociclib stabilizes this damaged microenvironment and the production of IL-6 and also improve reprogramming. We have found the barriers to reprogramming in vivo in the innate immune system. If we transiently deplete NK cells, we also exacerbate reprogramming, and also the nutritional requirements. If we provide vitamin B12, we boost the production of S-adenosyl methionine, the one-carbon metabolism, and this is a limiting factor for the reconfiguration of the chromatin, DNA methylation and histone methylation. The cells that are undergoing this process, they consume massive amounts of methionine, and for that they absolutely require vitamin B12 to such an extent that the normal vitamin B12 in the diet becomes insufficient or becomes limiting. If you increase it, you have more reprogramming. So we want to combine all these and eventually, hopefully, achieve this partial rejuvenation without the necessity of genetic factors. And with this, I'm going to stop. I suspect that I ran over time. And these are the people, these three persons are the persons who have done most of the work that I showed. And I am very thankful to them and looking forward for an interesting discussion with you. Thank you very much.
Thank you very much. Wonderful talk, Manuel. Wonderful work with lots of implementations, of course. In the end, maybe in 5 or 10 years, I will ask you a question. When do you think we could start the first human trial? But before doing so, you said that you first need the genetic or the Yamanaka factors first, and then you need damage. What kind of damage do you need? Because there are several types of damage, of course.
Yeah, we don't know.
Okay.
We have not distinguished different types of damage. I have to say that the Yamanaka factors themselves, they already are a damaging component. Many cells cannot survive to the expression of these factors. It is not known why some cells become damaged and some cells become reprogrammed. This is not known. But it's true that some cells are damaged. For example, MYC is a potent apoptotic factor. So perhaps in some cells, MYC dominates, triggers cell death, and the cell death triggers a damage in the tissue. And this is very prominent in the pancreas. The pancreas becomes very damaged, and the pancreas is a cascade. Pancreatic damage produces more pancreatic damage and more pancreatic damage. In other tissues, like in the lung, there is no damage, and what we did was to treat the lung with a genotoxic agent, with bleomycin. If we put bleomycin, this genotoxic agent synergizes very nicely with the Yamanaka factors. We know that in this damage, a key component is interleukin-6. The damage produces changes the microenvironment, releases cytokines, and we know that cytokine IL-6 is critical. If we deplete with antibodies in vivo IL-6, we reduce reprogramming a lot. If we do it in knockout mice for IL-6 receptor, we block reprogramming. IL-6 is absolutely critical.
So that's very interesting. And you showed that different organs are reacting very differently. And the pancreas seems to stand out. And I, as a clinician, if I hear pancreas, then I think about diabetes. So have you looked in your mice if also the glucose tolerance of these mice is better? Maybe it's too far stretched, but let me ask the question.
Yeah, no. It's an excellent question. We have studied that. The reprogramming that we observe, I didn't mention that, but it is in the acinar exocrine compartment. I show the acinar cells. Surprisingly, the endocrine cells, they remain completely oblivious to the pancreas. So you can reprogram the entire pancreas, exocrine pancreas, and the islets, they remain there normal. The same happens with cerulein. You can almost destroy the exocrine pancreas, and the endocrine compartment is very resilient.
Oh.
So yeah, no, we have not been able to rejuvenate the endocrine pancreas.
Okay, that's
work of Belmonte, Pisua Belmonte, they show rejuvenation in the endocrine compartment.
Yeah, but let me come back to that. Some cells react and some do not. So what is roughly the proportion that might also be organ specific, reacting and not reacting?
It's true. We don't know that. We are doing single cell RNA-seq to quantify that. We know that in the pancreas, a high percentage, it's difficult to say because the single cell RNA-seq also produces a distortion in the proportion of cells. But let's say 50% of the cells respond to this dedifferentiation because there are different stages here. The initial stages, many cells undergo dedifferentiation. Few cells go all the way to reprogramming. But probably for rejuvenation, for rejuvenation, we don't need to go all the way. We just need to induce dedifferentiation and then redifferentiation. That's all.
Yeah. But would that then mean that there's a proportion of cells which are undergoing the reprogramming, that you end up with a mosaic of cells? And what does that mean also with your head on as a cellular senescence researcher? So tell me, what does that mean, ending up in a mosaic of a lung?
These are super interesting questions that, of course, there is no answer for them. We are wondering the same. I would say that this is where the frontier is. You mentioned two aspects that are very important. So rejuvenation with the Yamanaka factors rejuvenates a subset of cells that we don't know if it is 50 or 70 or 10. We don't know. Probably by doing cycles of reprogramming, the proportion increases. Also, by doing cycles, probably the rejuvenated cells have the opportunity to rejuvenate the neighbors in a paracrine manner. This is a possibility. To answer this question, we need clocks that are single cell. And the field is now like crazy. I mean, we all are trying to find aging clocks that can measure the age of single cells. This doesn't exist yet. But that will answer your question about the mosaic and how this is interplaying young cells with old cells all together in the same organism. About the old cell, about the senescent cells. Senescent cells, so everything I said about reprogramming in vivo does not affect the senescent cells. Senescent cells, we publish that, and other people publish that, are resilient to reprogramming. the epigenetic configuration of senescent cells is resistant to the Yamanaka factors, not completely, but mostly. So we don't see a big impact immediately on the senescent cells. However, after cycles of reprogramming, perhaps the immune system becomes younger and the immune system is more efficient in removing the senescent cells. But immediately, reprogramming does not eliminate the senescent cells. Probably you need cycles.
Yeah.
Very good questions, Andrea.
Before really also going to the questions of the audience, I have one more question. So you treat these animals, the mice. How many of these mice really survive?
None.
No, because no.
Let me explain that. If we maintain doxycycline continuously, the degree of dedifferentiation in the intestine becomes incompatible with life. They cannot nurture, they cannot take the food. But this is continuous. So continuous doxycycline, they die.
Yep.
Everything I said today is 7 days of doxycycline and a low dose of doxycycline. You have to titrate the amount of doxycycline. It's 7 days. 7 days, we obtain this partial reprogramming that is reversible. If we maintain for 2 weeks and we stop the doxycycline at that point, the mice will develop teratomas. So
also
too
no good.
too much is death. If we go through the point of no return, the mice will develop teratomas eventually. To obtain the beneficial effects is just 1 week or 3 days. It's a small period of time.
Yeah. Wonderful. Hopefully we will get also to the interventions and the vitamin B12 studies, etc. you did, or the idea about supplementation it. But first, sorry.
This is the idea. The idea is that these pharmacological and nutritional interventions will not be able to go through the point of no return. They will be milder. They will produce a small change. And this is the idea behind. But we are far from that.
Yeah, but maybe we can come back to that. But now I would like to invite Guangzhou Ping to the stage. And I think there are lots of questions of the audience. So over to you.
Okay, thank you, Andrea. Prof. Serrano, thank you so much for this very interesting talk. Muchas gracias. There's so many questions from the floor. Okay, so we start from the most voted questions. Okay, number one from Sarah May. She asks, if the cell at their high fixed limit, the end stage, are they still able to be reprogrammed to embryonic stem cells? And does the telomeres or the telomerase reverse transcriptase play a part in the reprogramming?
Yes to both questions, but some explanations. When cells reach the Hayflick limit, they get very short telomeres, they activate senescence, they are very inefficiently reprogrammed. So if the cells are fully senescent, the reprogramming efficiency goes down orders of magnitude. Still, there are investigators that have published that some cells are able to reprogram. So it's not a zero, but it's orders of magnitude lower efficiency of reprogramming. I don't think that it will be very practical to rejuvenate senescent cells. I think that the idea with senescence is to eliminate them or to reduce the secretion of cytokines. Telomeres, yes, in the process of reprogramming, cells, actually at the point of no return, cells reactivate telomerase. Usually telomerase is silent in somatic cells. In the point of no return, telomerase is activated, telomeres are elongated, and the telomere clock is reset to embryonic state.
Well, I think I totally agree because senescence is a block reprogramming, right? So yeah, so the second question is from David Fan. Then he asked, the ability to reprogram organ system to a more youthful state can open up so many beneficial possibilities during aging. However, in your opinion, what would be some of the danger or challenges with this technology, and how long it takes to be overcome?
Well, yeah, I mean, I think that the main concern for me and for many colleagues is the tumorogenic potential of this process. The actual Yamanaka factors contain oncogenes. MYC is one of the most potent and famous oncogenes. KLF4 is another oncogene. And SOX2 is also oncogenic in different cancers. So we have here a cocktail of oncogenes. And I think I don't want to say pessimistic. I think that it will be difficult to fine-tune the genetic factors in such a way that they rejuvenate without any risk of cancer. I think that will be difficult. I'm not saying it will be impossible, but it will be difficult. And regulatory agencies will be very, very alert, I would say. I think that this is why the field is moving to reprogramming without the Yamanaka factors. Also because we don't need to go all the way to pluripotent cells. We know now that we only need to do half of the way, just to induce the differentiation and then redifferentiation. And perhaps we can achieve that with procedures that are not so pro-tumorogenic. So I think that this is my opinion, personal opinion. I think that it will be more promising non-genetic interventions.
So does that mean that a shot of IL-6 would be sufficient with a good vitamin B12 state?
We are doing those experiments now.
Yeah.
Of course, IL-6 has secondary effects. There is nothing that, well, vitamin B12 probably is very safe, but
I can confirm that, yes. Or it's safer than an IL-6 shot.
Yes.
Yes, thank you so much for that question. I think David Sinclair also tried OSK without the M to reprogram.
It's true, and this is very good, and it's already a very good step in the right direction. But still, SOX2 and KLF4 have tumorogenic activity lower, but still have. So I think it's a matter of concern. I'm not saying that it's not working. I'm saying that it's a concern.
Yes, totally agree. So you also mentioned that the OSKM silenced the transcription at the early stage of reprogramming, right? Is this actually a stress for the cells to undergo enforced revision? And also
to undergo what?
To force the cell to be reprogrammed. Is the stress necessary for the cell to undergo reprogram? And this is a question asked by Yai Zhu. So he also asked that also using OSKM for regeneration sometimes result in the loss of vital organ function. This may be reversible in some tissue by withdrawing OSKM. However, can all tissue automatically be redifferentiated upon OSKM withdrawal? Yeah.
Very good questions. Nobody has been able to dissect or to separate, to dissociate the differentiation from reprogramming. Always, the first thing that you need to do is to switch off the cell identity programs. So I think that is an intrinsic and necessary part of the program of reprogramming. You have to switch off cell identity genes. If not, you cannot induce reprogramming. And probably for rejuvenation, this is the only thing you need. Switch off the cell identity programs and allow the cells to activate again the same cell identity programs. This process of switching off and switching on could be, this is a speculation, could be at the basis of the epigenetic rejuvenation. It's a speculation. We have to think that when cells age, they acquire noise. Noise, this is known, noise in everything. Transcription becomes noisy. Splicing becomes noisy. Three-dimensional loops become noisy. All this has been published. Noise is part of the aging process. Who knows? Maybe the dramatic switching off of cell identity and then rebuilding everything again eliminates part of this noise. It's a speculation. Did I answer all the questions? There was another question.
Can all the cells be reversed?
All the cells.
Yeah.
We don't know that. We have explored only a few tissues. We have explored the pancreas, the intestine, stomach, kidney. But there are tissues that we have not explored. This is why we and others are now going in the direction of cell type specific reprogramming to address this question cell type by cell type. To what extent is reversible? Let me say that many cells are not reversible. Many cells are eliminated by the immune system or die by apoptosis. But a sufficient fraction to restore the tissue reverses. This is another pending question, to do lineage tracing and to quantify. Andrea was asking about that, to quantify how many cells undergo this process and what percentage of these cells go back and participate in the rejuvenation of the tissue. This is still unclear.
And how is, sorry to interrupt, but how is immunosenescence then interfering with it? So, because if cells are being recognized, being de-differentiated, are they then killed by a nice monocyte? And activate a macrophage?
Yeah, exactly. We know that natural killer cells recognize these de-differentiated cells as aberrant cells, and natural killer cells constitute a huge barrier for reprogramming. If you deplete NK cells, there is much more reprogramming in all tissues. The immune system is an important barrier. And we don't know, we have not studied how the immune system is rejuvenated. This could be very important for the clearance of senescent cells. Yeah, I'm sorry, there are so many open questions. I hope there are more people working on in vivo reprogramming. It's challenging and difficult, and it takes forever to do the experiments.
But that gives us the room to invite you again. But I think we have room for one or two more questions from the audience.
Okay. So one of the audience want to congratulate you and ask you, when are you moving to Cambridge? Is your friend from Madrid?
Yeah. I will be moving to this new company and laboratories called Altos. It's a stepwise process. I will start a double affiliation next year in May. I will start a double affiliation, and I will be full affiliation probably in 23 or so. So we have to wait until the laboratories are fully operational, including the mouse facility, which for me is very important.
Thank you so much. This concludes the question session. I think we have to ask you back with new results, because this is so promising. And what I learned is sometimes maybe to have noise cancelling for our cells, which are aging, and reprogram them. So thank you very much, and hopefully the listeners also, you enjoyed this very important and very nice session. Hope to see you again in the future. I would like to ask you to also give us feedback, because that's very important for us, and please use the chat function, the panelists and all attendees option, I must say. Next week, I will host Sarah Hilmar. She is a professor and a geriatrician and pharmacologist in Sydney in Australia. And I hope to see you on the 14th of October, same time, same room, but please register. And we conclude this session today with I would say, very wonderful video of an 87-year-old Iron nun. And I hope that you enjoy your next physical activity as this nun does. So take care.
Best known as the Iron Nun.
Iron Nun.
They call her the Iron Nun.
The Iron Nun.
Sister Booter is the oldest person ever to finish an Ironman triathlon, swimming 2.4 miles, biking 112 miles, and running a marathon.
When I was still in grammar school, maybe around 9 years old, my grandmother, a very insightful woman, opened the door and said, "Darling, what's the matter?" I said, "Well, I'm trying to figure out what I should do when I grow up." She said, "Darling, let's look at it this way. The past is dead and gone. The future is not the future until it gets here, and then it's the present moment. That is the only thing that you are responsible for." That made so much sense to me. It just took all the burden off my back. I have to remind myself that any kind of anxiety is developed by worrying about the future, of which none of us really have control over. So that brings me back to living in the present. I'll make the most of the here and now, right now.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.